Your blood type is not random. It is not a coincidence stamped on a hospital wristband. If you are type O positive, the DNA inside you carries the fingerprint of a war your ancestors fought and won, a war against a parasite that still kills a child every minute somewhere on Earth. In the year 1900, a scientist named Karl Landsteiner sat in a laboratory in Vienna, mixing blood samples from his colleagues, and watched something strange happen. For centuries before that moment, doctors attempting blood transfusions had been operating almost entirely blind, unable to explain why the same procedure could save one patient and kill another, with no obvious difference between them. Fundamental that it would go on to save more lives than almost any other discovery in modern medicine. He called the system ABO, and within it, he found 4 categories: A, B, AB, and the one that would turn out to be older, stranger, and more widespread than any of the others, the type we now call O. At first glance, O sounds like an absence, a blank space where something should be, and in a sense, that is exactly what it is. Every red blood cell in your body is covered in tiny molecular flags, sugar structures that sit on the surface like signposts, telling your immune system what belongs and what does not. People with type A blood carry one flag. People with type B carry another. People with AB carry both. And people with type O carry neither, because somewhere in deep human history, a single mutation broke the gene responsible for building those flags in the first place. That one deletion causes the cell's machinery to stop reading the instructions too early, like a sentence that gets cut off mid-word. The enzyme that should have been built never gets made. No flag goes up. The surface of the red blood cell stays bare, and that bare surface, that absence, is not a flaw. It is the entire reason type O exists, and the reason it may have once meant the difference between life and death for the people who carried it. Because here is the twist scientists were not expecting. That missing flag did not just leave your ancestors' blood cells empty, it left them invisible to something else entirely, something microscopic, something that had been hunting the human species since before recorded history even began. Every environment came with its own biological gauntlet, and the immune system had to adapt, or the population would not survive to pass on its genes. Blood type, it turns out, was one of the sharpest tools evolution had in that fight. A landmark genetic study published in the journal Scientific Reports set out to map exactly how blood types shifted as modern humans spread across the planet. The researchers were not just looking at living populations. They were comparing ancient DNA, genetic material pulled from bones and teeth that had been buried for tens of thousands of years, against the blood types carried by people alive today. And what they found was a story of constant pressure, constant adaptation, and constant war between the human immune system and the pathogens trying to break through it. One of the most startling discoveries in that research had nothing to do with modern humans at all. It involved Neanderthals. Buried in the genetic data was evidence that a rare blood group variant, one still found in a small number of people today, did not originate in modern humans at all. It came from Neanderthals, passed down through interbreeding that occurred when early humans pushed into Southeast Asia more than 65,000 years ago. That variant likely gave the humans who carried it a genuine survival advantage in unfamiliar territory, helping their immune systems recognize threats their bodies had never encountered before. It is a strange thing to sit with, the idea that a piece of Neanderthal biology might still be quietly working inside someone's bloodstream today, inherited across more than 2,000 generations. But the story of type O specifically leads somewhere even more dramatic, and it leads straight into one of the deadliest diseases humanity has ever faced: malaria. For most of human history, malaria was not a distant tropical illness. It was one of the single greatest killers of our species, a constant grinding pressure on populations living anywhere near mosquito-heavy wetlands, river basins, and warm climates. And within that pressure, researchers noticed something remarkable when they studied survival patterns across populations exposed to the most lethal strain of the parasite, Plasmodium falciparum. People with type O blood were surviving at meaningfully higher rates than people with type A or type B. The reason comes back to those molecular flags again. Malaria parasites are ruthlessly efficient. Once inside the bloodstream, they invade red blood cells and use them as hiding places, multiplying inside where the immune system struggles to reach them. But to spread from cell to cell efficiently, the parasite relies partly on those A and B surface markers, using them almost like handholds to help infected cells stick together and clump inside blood vessels, a process that can trigger the most severe and often fatal form of the disease, cerebral malaria. Type O blood, missing those markers entirely, gives the parasite far less to grip onto. Infected cells are less likely to clump. Blood flow to the brain is less likely to be blocked. And in a disease where the difference between mild illness and death often comes down to exactly that kind of blockage, a missing molecular flag turns into a genuine survival advantage. Researchers reviewing decades of data across malaria endemic regions in Africa and Asia have repeatedly confirmed this pattern. Populations tested in some of the highest transmission malaria zones on Earth show consistently lower rates of severe disease among people with type O blood compared to non-O blood types. It is one of the most well-documented examples scientists have of natural selection acting directly on the human immune system within a measurable, traceable window of evolutionary time. A Princeton population geneticist studying the broader picture of blood group evolution has described these systems as one of the strongest known targets of positive selection in human history, shaped again and again by the specific diseases ancient populations were forced to survive. This is almost certainly part of why type O blood is so common today in regions of the world that were historically hit hardest by malaria, and why the geographic map of blood type distribution still echoes, faintly but unmistakably, the ancient map of where malaria once ruled. In some indigenous populations, particularly in parts of Central and South America, type O blood appears at extraordinarily high frequencies, in some communities approaching nearly the entire population. A pattern researchers believe reflects both this ancient disease pressure and the narrow genetic bottlenecks these populations passed through during their migration and settlement history. Meanwhile, in parts of Central Asia and the Indian subcontinent, type B blood shows up far more frequently than almost anywhere else on Earth, another echo of a different regional disease landscape, shaping a different genetic outcome. If you were to lay a world map of blood type distribution over a world map of historical infectious disease pressure, the overlap is not perfect, but it is far too consistent to be coincidence. It is worth pausing here on just how much biology is actually packed into what most people think of as a single, simple label. The ABO system, the one determining whether you are A, B, AB, or O, is only one of dozens of separate blood group systems scientists have identified inside human blood. Researchers now recognize more than 40 distinct blood group systems in total, each governed by its own genetics, each shaped by its own evolutionary pressures, and each capable of triggering its own immune reactions if mismatched during a transfusion. Most of these systems stay invisible in ordinary life, only mattering in rare and specific medical circumstances, but they are a reminder that the four letters most of us learned as children, A, B, AB, and O, are really just the most visible layer of an enormously complex genetic landscape running beneath the surface of every human body on the planet. But evolution rarely hands out a gift without also attaching a cost, and type O blood is no exception. The same missing surface marker that made ancestors harder for malaria to exploit appears to have made them more vulnerable to something else entirely. A different kind of enemy spreading through contaminated water instead of mosquito bites. Cholera. Cholera devastated human populations for centuries, tearing through crowded cities, river settlements, and trade routes with terrifying speed, killing through catastrophic dehydration within hours of infection. And when scientists studied historical cholera outbreaks, they found the opposite pattern for malaria. People with type O blood were more likely to develop the severe, life-threatening form of the disease. The toxin, produced by the cholera bacterium, appears to interact more aggressively with cells that lack the A and B surface markers, intensifying the deadly fluid loss that defines the disease. The very same molecular absence that once protected your ancestors from malaria may have made them more fragile in the face of cholera. This trade-off is one of the clearest examples biology has to offer of a concept researchers call balanced polymorphism, the idea that no single blood type wins outright, because every genetic advantage comes bundled with a corresponding vulnerability. Nature did not design a perfect blood type. It designed a set of imperfect options, each one better suited to surviving certain threats and worse suited to surviving others, and different environments around the world quietly favored different combinations over tens of thousands of years. That is why blood type frequency is not evenly spread across the globe today. It is a living map of ancient disease pressure written into the DNA of every population on Earth. Cholera itself has a long and brutal history with the human species, sweeping through crowded port cities and river towns in devastating waves throughout the 18th and 19th centuries, killing so efficiently that entire neighborhoods could be emptied within days. Historical records from some of these outbreaks show mortality patterns that modern researchers have gone back and tried to reconstruct genetically, piecing together which populations were hit hardest, and cross-referencing that against the blood type. Distributions those regions carry today. The pattern that keeps emerging lines up with the laboratory evidence. Regions with historically intense cholera exposure show subtle shifts away from type O frequency compared to regions where cholera was less of a constant threat, as if the disease itself was quietly editing the genetic makeup of the populations it swept through, generation after generation, outbreak after outbreak. For a long time, this ancient story was treated as a fascinating but closed chapter, something interesting for anthropologists and evolutionary biologists to study, but not something with much bearing on modern life. That assumption has been quietly falling apart. In recent years, researchers have gained access to something earlier generations of scientists could only dream of: enormous medical databases containing genetic and health information on millions of real people. And when scientists in Sweden combed through health registry data covering more than 5 million individuals, cross-referencing a blood type against more than 1,000 different diseases, they confirmed something doctors had suspected for decades, but had never been able to prove at this scale. Blood type is not medically neutral. It is quietly correlated with real measurable health outcomes across dozens of conditions researchers had never even thought to check before. That massive study confirmed a pattern already suspected from smaller research, that people with type A blood face a higher risk of dangerous blood clots, while people with type O blood face a higher risk of the opposite problem, bleeding disorders. It also uncovered new connections nobody had documented before, including evidence that women with type O blood face a higher risk of pregnancy-induced hypertension, a serious condition that can threaten both mother and child if left unmanaged. The clotting difference in particular has become one of the most consistently reproduced findings in modern blood type research, and a 2025 systematic review pulled the picture into even sharper focus. Non-O blood types were found to carry substantially higher risk of pulmonary embolism, a dangerous blockage that occurs when a blood clot travels to the lungs, with researchers estimating that risk to be roughly 45% higher in people with type A or type AB blood compared to type O. The biological explanation traces back to a clotting protein called von Willebrand factor. People with non-O blood types tend to circulate significantly higher levels of this protein, which makes their blood more prone to forming clots. People with type O blood tend to run lower, which cuts clotting risk, but comes with its own trade-off, a somewhat higher tendency toward bleeding when the body is injured or during surgery. The same lower clotting tendency appears to work in type O's favor when it comes to the heart. Multiple studies tracking cardiovascular disease risk across large populations have found that people with type O blood tend to show the lowest overall risk of cardiovascular disease among the major blood groups, a pattern researchers link partly to naturally lower levels of inflammation and lower LDL cholesterol, the kind of cholesterol most strongly tied to artery blockages. Researchers are currently expanding this research into African and African-descended populations, specifically, a group historically underrepresented in blood type health research, to determine whether the same protective pattern holds true across different ancestries and environments. Cancer research has turned up another intriguing thread. Several studies have found that people with type O blood carry a meaningfully lower risk of developing stomach cancer compared to people with other blood types, a connection researchers believe may run through the same molecular surface markers involved in the malaria story, since those markers also influence how certain bacteria, including the ulcer-causing bacterium Helicobacter pylori, interact with the lining of the stomach. Digestive health research has surfaced a similar thread involving inflammatory bowel disease. Several studies out of Europe and Asia have looked specifically at whether blood type influences the risk or severity of conditions like Crohn's disease and ulcerative colitis, and while the findings have not always agreed with each other, a recurring pattern has shown up more than once, people with type O blood appearing somewhat less likely to develop Crohn's disease, and when they do develop it, tending toward a milder course of the condition compared to patients with other blood types. Researchers are careful to note that these effects are modest and not consistent across every study population, but the fact that the same protective signal keeps resurfacing across different countries and different research teams suggests there's a real, if still not fully understood, biological connection between the ABO gene and the way the gut's immune system behaves. But the picture is not universally protective, and this is where the story gets genuinely more complicated than most people assume. In critical care medicine, several recent studies have flagged type O blood as a potential risk factor rather than a protective one. Research out of Japan examining patients with severe trauma found that type O blood was associated with worse outcomes, including higher mortality, a pattern researchers linked back to that say, blood type and tracking outcomes closely, to investigate whether the same coagulation differences might make type O blood a risk factor for worse outcomes in overwhelming infection, precisely because the disease itself often involves dangerous disruptions to normal blood clotting throughout the entire body, not just at a single wound site. Even in surgery, researchers have started paying closer attention to blood type as a variable worth tracking. A recent study looking at patients undergoing liver surgery for cancer that had spread from the colon examined whether type O blood, with its lower levels of that same clotting protein, was associated with higher intraoperative blood. Pregnancy adds one more layer to this story, and it is one that touches an enormous number of families without most people ever realizing the underlying cause. When a parent with type O blood carries a baby who inherits type A or type B blood from the other parent, the parent's immune system can sometimes produce antibodies that cross the placenta and target the baby's red blood cells after birth. This condition, known as ABO hemolytic disease of the newborn, is usually mild, most often showing up as jaundice in the first days of life, and is typically resolved with simple monitoring or light therapy. It is far less dangerous than the more well-known Rh-related version of this condition, but it is a direct, everyday example of how deeply blood-type genetics reaches into ordinary human life, quietly shaping outcomes most parents never connect back to a molecular mutation tens of thousands of years old. And then there is the role type O blood plays in medicine today, a role that has nothing to do with ancient malaria, and everything to do with the fact that those same missing surface markers make this blood type uniquely valuable in the modern world. Because type O negative blood carries neither the A nor B marker and lacks the Rh marker as well, it can be given to patients of nearly any blood type in an emergency without the immune system, launching a dangerous attack against it. This is why type O blood is often called the universal donor type, why trauma centers and battlefield medics reach for it first when there's no time to test a patient's blood type before a life-saving transfusion, and why blood donation centers around the world consistently list type O as one of the most urgently needed types on their shelves. The Rh factor itself, the plus or minus sign attached to every blood type, adds one more layer to this story that most people never think to ask about. It was discovered decades after Landsteiner's original ABO work, when researchers realized that most humans carry an additional protein on the surface of their red blood cells, one they eventually named after the rhesus monkeys used in the early research that uncovered it. People who carry this protein are Rh positive. People who do not are Rh negative, a smaller group making up a minority of the global population, though that minority varies significantly from region to region. Combine the ABO system with the Rh system, and you get the 8 familiar, type O positive specifically, positive rather than negative, sits in an interesting middle position. It carries the malaria resistance and cardiovascular advantages tied to the missing A and B markers, but because it does carry the Rh protein, it does not share the extremely rare, universally compatible status of type O negative blood, the type most people picture when they hear the phrase universal donor. O positive is still broadly useful in transfusion medicine, compatible with any Rh positive patient regardless of their ABO type, which is one of the reasons it remains in constant demand at blood banks worldwide. But the true universal donor title belongs specifically to its negative counterpart, a distinction worth understanding if you have ever wondered why donation centers seem to ask for O blood with such particular urgency. Blood donation itself has quietly become one of the clearest real-world demonstrations of everything this ancient genetic story means for people living today. Hospitals depend on a constant, reliable supply of type O blood precisely because of situations where there is no time to run a full blood type match. A car accident victim arriving unconscious, a soldier wounded on a battlefield, a mother hemorrhaging during childbirth. In those moments, medical teams reach for type O blood because it is the one type least likely to trigger a dangerous immune reaction regardless of who receives it. Every single donation from a type O donor carries the potential to save a life in exactly the kind of emergency where there is no room for delay, which is part of why blood banks in nearly every country on Earth consistently report type O as one of their most urgently needed supplies, particularly during summer months and holiday periods when donation rates typically drop, even as demand from hospitals stays constant. That urgent, constant demand has pushed scientists toward one of the most genuinely futuristic breakthroughs in this entire field, and it is happening right now. Researchers have developed enzymes capable of stripping the A antigen directly off red blood cells and organ tissue, effectively converting type A blood into universal type O blood in A related technique has already been used to convert donor kidneys from type B to type O, demonstrating that this approach is not limited to blood cells alone, but may eventually extend to entire transplantable organs. If this technology continues to advance and eventually reaches widespread clinical use, it could fundamentally reshape the global blood supply, dramatically expanding the pool of organs and blood available for transplant and transfusion, and reducing the immunological barriers that currently cost lives every single day while patients wait for a compatible match. Researchers involved in this work have been careful to note that further studies are still needed to confirm the safety and long-term feasibility of these converted cells and tissues before they can be used routinely in hospitals. But the early results represent one of the most promising developments. Transfusion medicine has seen in decades, essentially using enzymes to rewrite, on demand, the same molecular code that natural selection spent tens of thousands of years shaping across entire populations. Step back from all of it, from the malaria resistance and the cholera vulnerability, from the clotting proteins and the cardiovascular data, from the Neanderthal DNA and the laboratory enzymes turning one blood type into another, and a single thread runs through the entire story. Type O blood is not simply the most common blood type on the planet because of random chance. It is common. Because, across an almost unimaginable stretch of human history, it repeatedly gave the people who carried it a fighting chance against some of the deadliest threats our species has ever faced. Every person walking around today with type O positive blood is, in a very real biological sense, carrying forward the outcome of that ancient war, encoded in a single broken gene that turned out to be far more than a flaw. Modern medicine is only just beginning to catch up with what evolution figured out tens of thousands of years ago, that this one blood type sits at a genuine crossroads of protection and vulnerability, of ancient advantage and modern risk, of ordinary biology and extraordinary history. It resisted one of the deadliest diseases our species has ever known, and in doing so, quietly shaped the genetic map of entire continents. It carried a cost in the form of cholera vulnerability that few people carrying it today have ever heard about. It lowers the risk of certain cancers and certain heart conditions, while raising the risk of dangerous bleeding after trauma, and, according to some research, worse outcomes during the most severe infections the body can face. It sits at the center of one of the most important tools modern medicine has ever developed, the universal donor system that saves countless lives every single day in emergency rooms around the world, and it now stands at the center of a genuinely futuristic scientific effort to rewrite blood type itself using laboratory enzymes. The next time a doctor draws blood or a donor card lists a simple letter and a plus sign, it is worth remembering that those two characters are not just a medical label. They are a message, passed down across more than 2,000 human generations, from ancestors who survived long enough to make sure the message reached you. Every clump that formed and every clump that did not, in laboratories and in ancient bloodstreams alike, was part of the same long chain of survival that ends with you reading these words right now. If this is the kind of hidden history you want more of, the story behind the biology written into your own body, make sure you stay close, because there is far more buried in your DNA than any of us were ever taught in school.